We are from Group 2. My name is Hon Kai Ting and my group mates are Farah Haslinda, Ku Jia-wen, Geraldine Lim and Hugh Huai Ming. So today, plant-wearable biosensors have gained a lot of attention due to its application in sustainable agricultural production. So in this video, we will be discussing its issue, significance, application, principle, and its improvements. So starting off with the first issue of plant-wearable biosensors is its lack of focus on plant biosensors. Because most of the wearable biosensors today have been applied for fitness and healthcare monitoring for human beings, there is an obvious lack of focus on plant sensors for sustainable agricultural production. And the second issue will be lack of comprehensive plant health monitoring. The current plant-wearable sensors focus more on monitoring plant growth, relative humidity, and plant diseases. There's a need for a more comprehensive assessment of plant health and there are more specific needs. And the third issue will be complex sample pretreatment. So the existing analytical techniques for pesticide detection still require a very complicated sample pretreatment, which kind of hinders rapid and non-destructive analysis. And the last issue will be need for wireless real-time information. There's a need for plant-wearable biosensors that can wirelessly transmit real-time information of pesticide residue to a smartphone for convenient monitoring, such as using the Bluetooth.
Now, let's move on to the significances of the plant-wearable biosensor. Firstly, plant-wearable biosensor allows real-time data transmission where the biosensor can send data instantly as it is collected, allowing immediate monitoring and decision-making based on the current pesticide level. The second significance is on the flexible and stretchable nature of the biosensor. It is able to adapt well to changes caused by environmental issues such as irregular crop surfaces and any other potential deformation. Thirdly, the biosensor is very selective. It can selectively capture parathion on crop surfaces after being modified with organophosphorus hydrolase, which also known as OPH, and equipped with biocompatible semi-solid electrolyte. Last but not least, the biosensor uses wireless connectivity and smartphone integration where it utilizes the wireless technology such as Bluetooth to establish a connection with smartphones and other devices.
Regarding the application, smart plant-wearable biosensor could be used for the in-situ analysis of pesticide residue on crops, contribute to the agriculture sector. A study has discovered that non-systemic pesticide after spraying will mostly remain on the surface of the crop until degradation. So, in this experiment, both spinach and apple have been selected to examine the power of the biosensor. Methyl parathion solution was sprayed on the leaves, and obviously, p-nitrophenol peaks were observed in the samples which contains the pesticide. By using this biosensor, we could more or less estimate the residue amount of the pesticides, which help us to have a thorough understanding towards the pesticide degradation process, as well as determine the reliable picking period for all the agricultural products.
Biosensor element, biomolecule immobilization, and transduction method are three major components that make up a biosensor. In context of a smart plant-wearable biosensor for in-situ pesticide analysis, the biosensor element is an enzyme that selectively captures and recognizes the target pesticide, namely organophosphorus hydrolase. Nafion, a biocompatible polymer widely used electrode modifier, is used to promote the immobilization of the organophosphorus hydrolase enzyme on the electrode surface. For the transduction method, the electrochemical behavior of the biosensor is measured using a handheld electrochemical workstation. The target molecule that can be detected using this plant-wearable electrochemical biosensor is organophosphorus pesticide, specifically methyl parathion. So, the construction of this plant-wearable electrochemical biosensor on crop surfaces goes like this. The polyimide film was patterned using the computer-controlled laser induction system to obtain the 3D porous laser-induced graphene-based serpentine 3 electrode. Next, the patterned polyimide film was covered with liquid PDMS and then heated. After that, the side of PDMS lead was peeled off from the polyimide film to obtain the flexible and stretchable lead-based electrode. Finally, the silver chloride ink was coated on the exposed reference electrode and heated until curing. The organophosphorus hydrolase was immobilized onto the sensing area of the electrode using naphthalene. In order to enhance the sensing behavior, the electrode was further decorated with gold nanoparticles and then equipped with biocompatible sensing electrolyte, for example, gelatin electrolyte. Now, the flexible and stretchable plant-wearable sensor, which can be well adapted to the irregular crop surfaces, can selectively capture and recognize the methyl parathion on crop surfaces. Square wave photometry is employed as the electrochemical measurement technique with specific parameters including a frequency of 8 Hz and an equilibration time of 30 seconds. Real-time information about pesticide residue could then be wirelessly transmitted to a smartphone using a handheld electrochemical workstation with Bluetooth. This analytical strategy eliminated the need for complicated sample pre-treatment and enabled rapid, non-destructive, and in-situ pesticide recognition on crop surfaces. The detection limit of this biosensor for in-situ analysis of methyl parathion is 0.01 micromolar, which can meet the demand for the detection of methyl parathion in vegetables and fruits announced by the European Commission.
In conclusion, in this work, a smart plant-wearable biosensor was developed for the in-situ detection of metaparathion on the surface of crops. A variable 3 electrode system can be easily patterned using the laser-induced graphene (LIG) technology on a commercial PI film by controlling two things, the laser power and the scan speed. By transferring to PDMS, a stretchable and flexible LIG electrode can be prepared, while still keeping the satisfying electrochemical response during the process of stretching and bending. This smart wearable biosensor was modified using OPH and equipped with gelatin electrolyte, which can help it to attach on the surface of agricultural products, such as the leaves of spinach and the fruits of apple, which allows two things, the real-time and also the in-situ analysis of metaparathion on the electrochemical determination of the corresponding hydrolysis product. Thus, this work is very vital for the future development of our agricultural sector. Moving on to the future works or improvements that can be made to make this smart plant-wearable biosensor better. First of all, we should focus more on the influence of environmental conditions such as the temperature and the humidity, and also the various plant interfaces on this smart plant-wearable biosensor. Secondly, we should focus more on developing a more stable recognition element and also exploring this area in order to satisfy a long-time monitoring during the whole plant growth. This is so that the result will be more constant and will be more ideal for future researchers. Thank you.